Well, hello, everyone, and thank you for joining us today for our discussion with the scientist. My name is Dalton Dietrich, and today I have the pleasure of talking to Dr. Nagi Ayad, who's an associate professor in the Department of Neurological Surgery, a very important member of the Miami Project to Cure Paralysis. In fact, one of our newest members. So, Nagi, welcome to the Miami Project. We're really excited about your work that we'll talk about today.
Thank you, Dalton. It's wonderful to be here.
Great. So let's just talk about your laboratory first. You've been working on many types of questions using molecular techniques. So what is the focus of your research within the university as well as the Miami Project?
Thank you. I think my laboratory has two components. It has a basic science component and a translational component. So for the basic science component, we really want to understand cell cycle transition in the developing brain and the developing nervous system. So I've studied the cerebellum for a long time, and the cerebellum is very interesting because it develops after birth, and there a certain progenitor cell called the granule cell progenitor divides a certain number of times and then exits the cell cycle to become a neuron. And that process is not understood fundamentally, how that process is irreversible. So this is important for both neurobiology, but also for cancer, because when the cell division goes awry and the cell continues to divide, it doesn't become a neuron, you have medulloblastoma, the most common pediatric brain tumor. So we've been trying to understand those processes. We've been looking at kinase pathways and epigenetic pathways. So kinases are enzymes that modify proteins, and they've really become great targets for drugs in different cancers. Epigenetic pathways are also great targets, newer targets in cancer. And what epigenetic pathways do is they modify chromatin. So in the developing nervous system, we found that in the cerebellum, you need certain kinases and certain epigenetic pathways to be turned on to get proper development. And if that doesn't happen, then you get medulloblastoma. Right. So that's the basic science aspect. I feel that informs the translational aspect, because we find very selective small molecules to target these proteins. And we work with clinicians for both medulloblastoma and glioblastoma and see if we can use compounds that get into the brain in clinical trials. And interestingly, these targets are also targets in spinal cord injury and traumatic brain injury. So sort of shifting gears, the same pathways that are involved in cancer many times are involved in inflammation. So we've been working with several groups at the Miami Project to see if the same molecules can be used in preclinical settings for spinal cord injury and traumatic brain injury.
Yes, we talk about that a lot, how our knowledge in one particular field can overlap with another completely different field, but sure enough, some of the mechanisms are still there. I think many people know that the Miami Project is uniquely associated with the Department of Neurological Surgery at the University of Miami, and also associated heavily with the Sylvester Cancer Center. So there we're doing a lot of operations targeting brain tumors, and you and your colleagues have found how heterogeneous these brain tumors are. So it's almost specialized medicine in which you're trying to understand the genetics of a particular type of brain cancer, and then trying to gear your therapies against that particular genetic population of cells, if you will.
Right. So we developed a computational pipeline basically to deal with the issue of heterogeneity in glioblastoma. So every tumor is different. So how do you find a drug or drug combination that's specific for each patient? So we worked with Stefan Schur, a computational biologist and a friend of mine, to develop a computational program that basically says for this kind of tumor, these kind of compounds should work the best. And That's for what's called intertumor heterogeneity. Every tumor is different. We do a lot of what's called single cell RNA sequencing to look at within the tumor how it's different. So you don't just have tumor cells, you have myeloid cells, other cells within a tumor. And then that's our next big challenge, is how to find compounds to target different parts of the tumor. Again, that translates into our interest in spinal cord injury that we've been working with Dr. Jay Lee's lab. Again, looking at single cell RNA sequencing information that they have from the spinal cord after injury, and also looking at different cells within the spinal cord to see if we can target different cells using our computational pipeline.
That's what I was so excited about bringing you to the project, is the new tools and technologies that you have been working with. And yes, even though myself studying inflammatory cells, getting down to the single cell level to actually really distinguish these different types of populations of inflammatory cells that we used to group according to their, their morphology and some crude biochemical characteristics. Now they're actually more complex, and that's going to again help us at the end of the day to develop new drugs and therapies. So that's very exciting. I think you have a very interesting background in terms of where you come from, where you worked, and things like that. Can you tell us about that and how that's brought with you new ways in which looking at translational research and developing new therapeutic interventions?
Yes, thank you. So yeah, I was trained as a cell biologist, really looking at fundamental mechanisms first as a graduate student at Yale in the cell biology department, looking at fundamental mechanisms of cell division during endocytosis. And then I did a postdoctoral fellowship at Harvard, again, looking at fundamental mechanisms of cell division. My first faculty position was at the Scripps Research Institute in Florida. And there I was really introduced to high throughput approaches to study biology on a genome-wide level. So I did a lot of what are called high throughput screens, identifying molecules that affect different pathways during cell division. And that's how really I found some compounds that were effective in preclinical models of medulloblastoma and glioblastoma. The same molecules we're interested in spinal cord injury. So that kind of approach has brought me to my current view of how to approach science, where I'm interested in both the cell biology of the problem, so going back to the single cell issue. Every cell is different, and how do we understand that molecularly? But also globally, how do we use high throughput approaches to integrate information and get some answers to our basic science questions, but also preclinical and clinical questions. Because going forward, we can't think of one pathway that's going to be altered when we put on a compound. We have to think in a systems biology fashion, in an integrative fashion, how the whole system is going to be affected.
In addition to academia, you work very closely in some of your studies with industry. How has that helped you maybe move some of the therapies forward?
Yes, thank you. So yeah, we've worked with various companies who had interest in the targets that we were working on for other indications. They had compounds that were brain penetrant, and we worked with them to try them in our preclinical models of medulloblastoma and glioblastoma. Because of the computational work, now several companies are coming to us and saying, in clinical trials, can we find a way to work together to predict patient stratification using methodologies that we've developed along with Dr. Schurer, to see if we can stratify patients, which patients should be given which drugs in their pipeline.
Very good. Yeah, you went over very quickly the epigenetic studies that you're doing. I've been very interested in that for many, many years. We think about how the environment affects someone's health. And we're now knowing more and more about epigenetic modifications. And of course, much of your work is actually directed toward trying to target those epigenetic processes. So I think, again, opening up a whole new way to think about science, pathophysiology, and drug treatment. So I'm really excited that you're here. You're working with people doing spinal cord injury, traumatic brain injury, brain tumors, and that's very exciting to have you associated with the project and our program. So anyway, it's been great talking to you today. Thank you very much.
Great talking to you.
As you can see, Nagi's outside. We're still in this COVID-19 pandemic, and so we're actually having to continue to work remotely. But how is your lab going? Now that I brought it up, how is your lab research progressing while we're trying to, limit the opportunities to pick up viruses and things of this nature through and social distancing, making sure we're not, interacting too much, but at the same time, very interested in moving our science forward.
Yeah, so it's been an interesting transition. So number one, I've been worried about the safety of people that work in the lab. So we made sure that we minimize people coming in. But because we do a lot of computational work, this time has allowed us to basically take a pause and say, can we compare data sets that we have with what's out there to make certain predictions? So that's been helpful in a way. So if there's anything that's positive that's come out of this, is that this pause allowed us to actually expand our data sets and understand things a little better. We finished reviews and obviously finishing grants. We hope to be back very soon and continue with our animal studies, preclinical studies. And many partners in industry are still calling us to see if we can still make predictions for clinical trials that they're still planning. So those studies are ongoing.
Fantastic. Well, again, Nagi, thank you very much for spending time with us. Congratulations on all the work you're doing, and look forward to seeing your work progress, and we'll bring you back to talk about your research efforts in the near future. Have a great day. Thank you.
Thank you so much.